While Tor is setting up, maybe I can introduce him. I used to know Tor while he was in Boulder, but now he's in Dartmouth. And Tor has been really interested, as far as I know, in the study of pain, but really been going at it, pushing as much as possible the methods. And by methods, I mean something very general. So using imaging and other things also. And so that's a little introduction, because his title doesn't really convey the fact that he comes really with an eye also on applications. Thanks a lot, Tor, for being here. The floor is yours.
Thanks, Carl. Really glad to be here and talk to you all. Let me know if you can't see my screen here, the title screen. And I suppose due to my screen setup, I can't see any of you or your comments. But if there are comments or questions, then just feel free to break in, or Carl, you can if you'd like. Okay. So glad to be here, and just wanted to say a few things about and talk to y'all about how it used to be and what a long way we've come from the old days. And I don't mean the good old days, really. So when I started graduate school, there are several things that would really seem crazy right now. First of all, sample size. The most we ever thought of scanning was 10 subjects. 20 subjects seems like a huge study. It never seemed like this was going to change due to the cost of imaging. But I remember the first time I heard a talk where somebody tested 100 subjects, and it just seemed insane. Code. Data processing was done with a graphic interface in SPM or in Voxbow, which some of you may never have heard of because it doesn't exist anymore. SPM did not have a batch mode. Python was just a snake. FSL didn't exist at all. You could write your own code in IDL or MATLAB or S+. R wasn't even released until 2000, which was after I, was an advanced graduate student. The GLM was a new invention in analyzing fMRI data. People would treat participant as a fixed effect, and they analyzed brain data as though they're one big subject, which is probably related to the fact that you only have 6 subjects in your experiment. People analyze these peristimulus time histograms using f-tests, which is really a not very powerful way to analyze data without accounting for the trial history or anything like that. There was really very little way of visualizing data. So I started coding in MATLAB and Shell, partly because I needed a way to look at my data, when I didn't find results that popped up in the maps. But there really is still pretty limited ways of doing that, and they're getting a lot better. There were very few people who were using or passionate about using multivariate models. Mainly, we treated every bit of brain as a separate bit to be analyzed separately. And this was really a carryover from a tradition in neuropsychology where one brain region was supposed to encapsulate a mental function or module. And the field was early on built on looking where the light is in terms of areas where you have focal brain damage and some deficit. But as we've learned since, the brain doesn't really work that way. It was very common to smooth the heck out of brains to satisfy statistical assumptions and increase anatomical alignment, but thereby reducing all the anatomical specificity and signal. It was extremely common practice, even by very smart people and well-meaning people, to make reverse inferences. So I remember these conversations where, if you're doing a face task and the hippocampus is activated, you think, there must be memory involved. If the amygdala is activated by viewing other race faces, they must be threatening faces. This must be an indicator for threat. And underlying some of these problems is the fact that there was no ground truth. Because if all you're trying to do is model brain activity, we don't know what the brain activity is supposed to look like. So brain decoding didn't exist. And the idea of making predictions about objective class labels or performance measures or external variables really didn't exist. There was a lack of replication. There was no reproducibility or replication crisis, and there were no standards for what counted as a replication. There was a lack of consensus. So when I started graduate school, there were no methods for quantitative meta-analyses or summaries across different studies to see what's consistent, across the various studies. Data sharing was nearly unheard of, and I remember the fMRI data center at Dartmouth was a really early effort, and this was like 4 terabytes, which seemed like a crazy amount of storage space that you could never do anything with, and it wasn't standardized in terms of the metadata and so forth. And in spite of all these problems, it was really common to talk about blobs on brains as revealing the mechanisms underlying mental representations. So the old days, are behind us. But as a result, people really did compare imaging to phrenology, or maybe like kind of phrenology. All right, so here's my list, by the way, what I just said. You can see it all on YouTube, but 12 problems. And so this is my first talk where it's the I'm an old guy talk. I'm trying it on out for size here. But I'm also a happy old guy because things have really changed. So this was then, and this is now. We have massive studies. We have great tools with great coders and reproducible code. I'm giving green to the good things. Version, control. We have version control gets to yellow. I think our coding practices are getting a lot better. We have mostly better statistical models. We have more visualization of data and more work. And just to be honest, I think very few people check statistical assumptions or really visualize their data before running them through pipelines and models. So I give that one a red. It's not all roses, I suppose. We have predictive models, that I'm going to talk more about, and connectivity models, which model brain interactions. We have pattern information at high-res 7T studies. We're doing a 7T study with over 100 people now, and layer fMRI. We have a little bit less reverse inference. We have falsifiable predictive models, which are tied into this idea of using machine learning that I'll talk about. We still have problems with replication, but we do have meta-analyses that can really demonstrate remarkable consistency in certain basic phenomena across fields. We have massive data sharing, and we still make a lot of claims about mechanisms for various reasons. Here's a little table I compiled of some of these large-scale studies, including some that people in this group are really working on, Imogen and the HCP, of course, that Joe just talked about. And it's amazing. I can't keep up with all the stuff that's being shared. And even our 100 terabytes of storage space in my lab is going to be obsolete soon. And thank you to the UK Biobank team for this visualization of what 100,000 brains look like. Here's one other example that I picked out because it was came up recently on Twitter, as many of us find out new preprints and things from. And this is from Vince Calhoun's lab. It's just an example project. They called Neuromark. They called it Neuromark, and it's an automated ICA-based prediction engine, essentially. And they've got this preprint where they developed this on a large sample, something like 12,000 brains. They're predicting external outcomes, including cross-diagnostic, categories, which is one of the holy grails in, psychiatry and neurology. And the idea of what they have here is that it allows one to make structure-function mappings that vary across individuals. So you don't need to assume that the same brain feature or bit is the same across every subject. And there's a family of techniques that are exciting that can help to address those kinds of things. Shared model, and so lots of great. And this is in the kinds of collaborative multidisciplinary samples we have. And they also started a competition, a Kaggle competition, which is a really interesting development too, predicting, again, objective outcomes and letting teams compete to try to develop good models. So there it is if you want to join. And thanks to Vince for sending me these slides when I asked him. So yes, we've come a long way from that. I think it's really important to keep that in mind. as we think about what the future holds. And this has only been a few short years. I mean, I'm not really that old, like I said. Okay, so that was the road behind. Let me talk a little bit about the benefits of some predictive models, and I'll come up with an example from our lab. As many of you know, or everybody knows, traditional brain mapping involves taking some task-related measures or external variables and relating them to bits of the brain one at a time. And that creates a brain map of which bits of the brain respond to which different kinds of stimuli. But it doesn't tell us about how brain areas work together to model outcomes. And so with a predictive map or a predictive model, the general idea is to reverse that equation. And now the external variables become the outcomes, one or more. In the simplest case, it's one. And the brain variables are allowed to jointly predict the outcome and interact, potentially in arbitrarily complex ways, really. And the simplest one is a linear predictive model, and that's what we've mainly been using until we need to get more complicated because the data tell us we need to. And another feature of this is that once you develop that model, you can test that prospectively on new samples. And ideally, if you're making predictions about individual people, there would be new individual people that you can test, bring in de novo that you've never tested the model on before. And those can be within person. You can have multiple images within a person, and you're predicting continuous outcomes within a person or between people. And you can also, of course, use the develop classifier models or even use continuous models for classification. So this idea of prospective testing is another feature. And what that does is it makes the models falsifiable so that you can say in a new data set across laboratories, yes or no, this model works. And you can say which models work better. So I want to make this one point here, that predictive mapping serves both predictive and explanatory inferential goals. People often think of prediction and explanation as opposing things or different things, and for partly good reasons. But I think there's also a lot of common ground. So let's say we have our two doctors, Dr. A and Dr. B, and Dr. A is an applied scientist, and she's looking for biomarkers, maybe biomarkers for pain. And that's an objectively measured process that indicates some underlying pathology that we can't see. It might serve as a surrogate or other outcomes that we can't trust what people are telling us, or they can't tell us, or even risk factors that don't yet exist. Okay, so our Dr. A is interested in this applied, use of biomarkers. And by the way, the FDA has defined a really nice taxonomy of multiple types of biomarkers, at least 8 different categories of biomarkers, and what they can be used for. So she might want to know this at the end of the day, is this baby in? And if you don't have biological markers that are grounded in physiological processes and in brain processes, then your theories are really based on shifting sands, right? There's really no ground truth. And so the status quo in medicine was for thousands of years to believe that disorders were caused by imbalances between 4 humors. And this goes back at least to Galen, or probably before. And you still see this today. It's a kind of magical thinking, but of course, without biological measures, you can't falsify that or test it. And on the other side, Dr. B is a basic scientist. So he's interested in mental representations and their corresponding brain representations, which I'll just define here in a working way as a causal obligatory physical basis for a mental experience or any kind of other information. So when you stimulate that brain pattern or thing, then you have the response. When you block it, you don't have the response. So Dr. B might ask, does observing other people in pain activate shared sensory representations with my own pain system? And that might be important because maybe we evolved social representations on top of our own systems for representing value and experiences and physical harm. So here's this common ground between prediction and explanation. And I would say that if you have one without the other, it's not going to be a very satisfying answer. So prediction without explanation is a black box. You don't know when can you trust it. When is it being unfair? When is it using people's zip codes to make insurance decisions? And capitalizing on variables like race that you shouldn't consider. If you have explanation without prediction, this is an unfalsifiable explanation because there's no testable theory. So you never know, like the four humors, if the explanation is right or if it's useful. So the sweet spot, I think, is models that can develop aspects of both. So it's explanation that's supported by testable, falsifiable prediction. And I think this is the tradition that made physics so great and is being imported into biological sciences. And so both of our doctors here are interested in somewhat different things, but they're really interested in measuring the brain representations that underlie constructs in a way that enables valid measurement models. So a construct is really a human category, like some class of pains, or anger, or intelligence, which is some class of performance measures with some predictive validity in the world, and so forth. And in order to come up with those biomarkers or representations, there's a common set of empirical criteria that we've been thinking about for a few years now and writing about. For example, it ought to be testable. You can put a new person in that model and test it. It should be sensitive to the class of phenomena, the construct. It should be specific. It should be robust to perturbation and variation in the task or the setting or the scanner or whatever the irrelevant variables are. It ought to be generalizable to other instances of that same category or construct. If it's pain, multiple types of pain that are all that would fit within that construct. It should be interpretable, so we know when it's going wrong. It should be tractable and pragmatic to estimate at least the models and procedures that are more tractable are going to be the ones that are easiest to pick up and spread and use, so they will be the ones that survive. So predictive mapping offers a way forward that I think is really important. I think this is why it's taking off, and this is why the machine learning literature in brain has been so exciting. There's three things it does for us. Number one, it reduces analysis flexibility. If we can pre-specify a model and test that exact same model, that moves us towards strong inference and falsifiable theories about brain to outcome or performance mapping. Number two, it capitalizes on distributed codes, which can massively increase the effect sizes in terms of our predictive ability. And number three, model sharing and prospective tests can help us to establish generalizability and even develop models that are the most generalizable. So we're doing something that's very much at the heart of this construct validation process. And the rest of the examples that I'll show are examples that sort of bear on this. And I'll talk a little bit about these three things briefly. First of all, it's now come to light that if you are allowed to have flexibility in your analysis pipeline, and which variables you include, in which cases, and how you transform those variables, and how you extract features, and in a brain map, which voxels are significant or which features count as replications of an effect or as positive evidence for an effect, then you can massively overfit pretty much any data set, and you can end up with conclusions that are really not falsifiable, right? So lots of things can counter support for your theory. And here's an example from a recent paper from Wani Wu's lab, who is doing some great work in this area. And it's an example of social rejection from some data from our lab. And the idea is this: you find, you do this study in 30 people, and this is real data, and you find that there's this activation somewhere here in the anterior cingulate cortex. You say, aha, the anterior cingulate is activated. Now let's replicate that in a separate sample. And now I find this map, and you can see, and you can see with and you go, wow, the anterior cingulate's activated. That's a direct replication. Wonderful. Look how replicable neuroimaging is. But there are some problems with that kind of inference. And the problems really go back to the heart of how we aggregate information into evidence for our theories. So if you think about it, there's really no ground truth for how close is close enough to count as a replication. So in this case, there are no voxels above the statistical threshold in common, The peaks are 4 centimeters apart, which is a huge swath of brain space. And the patterns of activity are uncorrelated. So if pattern information is important, these responses are not the same and they're not replicable. And what Wani and his lab did is unpack this in great detail and provide some recommendations both at the pattern level and at the voxel, peak voxel interpretation level. And a long story short, most of the studies, even most of the studies that claim to directly replicate another study in the imaging literature, don't provide enough quantitative information to assess that replication. And when they do, their findings are not so similar. So, for example, the average distance apart is greater than the diameter of the entire amygdala itself. Here's another example that some of you might know about, and I've included this because it hit the presses at Nature yesterday, and it was by my current postdoc, Rotem Botnik-Nezer, but I was not involved in this study, sadly, at all, so I won't be putting Nature on my CV here. But the idea is what they did is took a data set on decision making, and they framed 9 hypotheses, and they had a bunch of teams compete, 70 teams to compete. And the punchline here is that on many of the questions, the teams with the same data set just didn't agree. They don't produce the same decisions. But these are decisions, and that's what's in the news. But these are decisions about which effects are significant, and that's what you'd see in red and green up here at the top. So there's a lot of disagreement about whether certain regions are activated or not in these different tests. But I love this figure because what it shows is if you look at the spatial patterns that are produced, all of this sort of red area is a positive intercorrelation in the spatial patterns that are found across teams. So they're very similar, actually. And you can also see that there's a subset of teams here where something wacky happened, and they're another group where something went wrong, they think, some outliers, and then some sort of other random stuff. But there's this consensus of actually very similar maps. What's happening is you're taking these noisy maps, which are very similar, adding a little noise, and you threshold it, and then different regions pop up. So it's really the problem is with the way we're doing inference and thresholding our maps, not with the reproducibility of brain maps themselves. And my thesis here is that if we use the pattern level and make predictions based on distributed patterns, the stability and the reproducibility is going to be dramatically higher. And that's what we've been finding. And why might it be higher? Well, it's in part because
is red in one mouse,
there it is.
Green in another.
The way the brain represents information is distributed. So this is an example, this rainbow example, where the cells are all randomly labeled. But the idea is that if you look at cell by cell, these populations, there are local ensembles of cells that respond to particular classes of events, and they're intermixed with other types of cells that respond to different classes of events. And these local ensembles are connected into circuits across regions. So you'll have a circuit in the hypothalamus and ensembles in the hypothalamus which communicate with ensembles in the PAG and ensembles in the accumbens and so forth in distributed circuits to accomplish functions. And so, there's an axiom that the best model will match the data generating process. Well, what's the data generating process for things like perceiving a stimulus even, or judging whether you like ice cream, or any other process that we might think of? How much pain do you feel right now? And across fields, there's really a growing amount of evidence in all these papers here that the operative information is distributed in neural populations within and across regions. And this has really profound effects for how we think about brain similarity as related to psychological similarity or performance similarity. And I like this quote from the Puget review. As in any good democracy, individual neurons count for little. It's population activity that matters. So one neuron might be activated in 2 different tasks, but as a part of 2 different population patterns. So here's some rules from this literature, basically, that cells with different functions are intermixed, individual neurons are noisy and not closely linked to behavior. And usually what people do is pick out the highlights of the ones that are, but there's interesting demonstrations that even without those, the rest of the neurons that aren't so strongly category selective or active are really the important ones. And of course, distributions across brain areas. And the work on using essentially modern opto and chemogenetic tools to outline circuits across regions is really showing that in very beautiful ways, where you can tag and reactivate a particular ensemble, like here in the MCC, with particular projection, in this case to the posterior insole of a rodent, which serves a particular function in this case, enhancing pain after a nerve injury at the central level. And here's some other work from Steve Ramirez and Tonegawa, very famous work on memory engrams, tagging and reactivating hippocampal ensembles, and it's happening more and more, so it's very exciting. So that's what we ought to be trying to capture, is those distributed patterns. And finally, if we're developing models that are predictive across individuals, then we can really use those to demonstrate and test construct validity. So this is where I'm going to turn to the example from our lab. And I picked this one. This is a pain signature, really one of the first predictive models that we developed in my lab. And I'm choosing it to talk about here because we've just been trying to, hammer away at validating it and testing it in different settings. And so I think that itself provides kind of an interesting example of the principles that I've just told you about. So we take a training sample, we use a penalized regression model, lasso PCR. Actually, maybe you don't even need the lasso. And we're trying to predict continuous pain ratings within and across person, develop a linear map, which is the best predictive weights. So it's a probably one of the most basic kinds of setups, algorithms that you could imagine, but it's also possibly one of the most interpretable, I'll argue. And so we called this the neurologic pain signature. Why did we give it a name? First of all, because the journal editors told us to, and they told us what to call it, so there it is. But secondly, we wanted to develop something that we can reproduce and use across different data sets without changing the model. And so you got to name something in order to reuse the exact same thing. And the model includes activity in regions that we know and care about that are related to pain and nociception and can be validated in invasive studies in animals and in humans, by and large. And so that's a sensible pattern, which is nice. And I think the key thing here is that we have distributed information across regions, and we think that this local information also is likely to be important in many cases. So, two principles of how we do this. One is the model is designed to generalize to new individuals. And we've tested this against individualized models where every person gets their own model, which has been kind of much more the status quo in the MVPA literature. And there's really some nice features. And the main one I'll point out is that they provide flexible and open-ended tests on additional data sets, additional participants. Secondly, we're trying to capture things that are common across people. And so there's much less room for idiosyncratic influences of confounds that might influence different individuals in different ways. And that's an issue with the single person prediction literature. And as always, I think you guys have been doing this better, but we're striving to to follow your lead, writing simple modular code, having really readable scripts for interactive analysis. So in the tools that we develop, you can load an object in with a command and then do a standard, cross-validated stratified prediction with another command, and then use one of a number of display options to render it on surfaces and plot the response. Forth. So we really want to build the script out of these very simple commands, which do things that we do often. So we can apply signatures to new test data from new individuals and new samples. And this is one example of doing that with 1 degree increments in noxious heat, low, medium, high, 46, 47, 48 degrees Celsius on the arm. And that gives us a continuous readout that we can turn into a classifier and ask, if we didn't know ahead of time, would we guess correctly which is the high and the low pain based on the brain? And we would get it right most of the time here. Here's another example. We can apply it to new data sets with different kinds of conditions. Now 6, stimulus intensities that range from non-painful to painful. And we can ask, how strong is the model response when it's in the painful range, which is quite strong, and in the non-painful range, which is not so strong? And what's its function across those, the different stimulus intensities? And what happens when you predict the subjective pain controlling for stimulus intensity and vice versa, or matching on stimulus intensity and so forth? So you can really interrogate that model and test it in various ways. And that's all a function of being able to have a fixed model that we can simply obtain a response from. And so then we can test this generalization in broader ways. Here's another example where I love this project because it really is a collaboration across many different labs who all shared their person-level data of pain and placebo effects. So we've got 600 people in the sample, multiple types of painful stimulation. And you can see the studies here are going down, each of 20 studies, and the smaller little dots are individual participants. And the shift to the right indicates that there is a positive response in this particular marker, NPS, to painful stimulation. And we can then quantify the effect size in an unbiased way because all of these studies are out of sample. We didn't fit the model again or do anything. And we didn't even sort of change the pre-processing procedures to match what we thought should be the case. We just took the studies kind of as we found them as published. So it's probably not as well as you could do, if you really standardize the processing. But what it does give you a sense of is what the effect sizes are for a sort of a broad generalization across different use cases. And here the effect sizes are really much, much larger than we've ever seen in a valid univariate test of a single brain region. And that's really a test of generalization, and we can think about with these models establishing several different kinds of generalization gradients, in what kind of stimulus or task does this respond to? Is it a very specific task or like a particular kind of pain here? Or is it a similar class of pains or tasks, whatever they are, or a very broad set or very diverse exemplars of a construct I'm interested in, like pain, or working memory? Does it apply to one person only, which is most predictive maps, or does it generalize across individuals or multiple samples or heterogeneous populations? And of course, if we're going to develop measures for psychiatry and that are useful clinically and translationally, we have to get all the way out to these heterogeneous populations, or at least know exactly whom it's going to apply to and who it's not. And what about context? Does it apply in a narrow task context or in very broad context? And so with this kind of model, we're able to explore all of these different dimensions. We're also able to test its specificity relative to a number of other conditions. And this is really an open-ended process because you can always come up with something where you say, well, I think this thing is similar to pain and produces similar kinds of brain activity, so it's confusable. So we can say, great, let's test it. So if I have this putative generalizable model here, our Dr. A is asking, what is this thing really measuring? Is it really measuring pain? Well, if so, it should generalize across multiple instances of very different types of pains within a certain category, like evoked sensory pain, which is what I just showed you. And Dr. B is asking, which parts of the brain are important? And I'll get to some ways of making some inferences on that pretty soon. And both of them might be interested in what is this going to respond to and what does it not respond to? So is there discriminability from other constructs? So one example of that kind of discriminability is comparing somatic pain to vicarious pain, observing other people in pain. So here we have different levels of somatic pain on upper and lower limb, and different levels, pre-selected images with particular levels of vicarious pain on upper and lower limb. So this gives us a 2x3 within person test of both replication and generalizability across body sites and tracking intensity, and it gives us a test of whether our patterns are specific. And both of these produce very similar subjective responses across both body sites and across intensity levels. So we can see, we have some really nice similar subjective aversiveness responses. And now we can compare the brains. We can look at our NPS measure, and what we find is it generalizes across hand and foot. Doesn't mean we can't tell hand and foot apart, right? But it tracks intensity in a generalizable way across body sites. And the blue bars here show that there's no response that looks anything like that to observed pain in other people. And now we can ask, well, maybe observed pain is just a really bad signal. It's a weak signal. It doesn't, but it does activate virtually all of the same gross anatomical regions. And we can run the same prediction process on the vicarious pain stimuli and predicting the subjective intensity of vicarious pain in response to others. And we get a different looking pattern. And that pattern tracks across body sites the intensity of vicarious pain, but there's no response to somatic pain. So that dissociation is called separate modifiability after some work in 2001 by Saul Sternberg. And that's a strong way of dissociating two constructs in terms of what they represent. And if you're interested in the paper, we did a zillion other analyses of individual regions and searchlights and lots of other stuff. Idiographic predictions where every subject gets a different pattern, and there's very little benefit to that as well. So this basic story really holds up, and if you beat on the data in lots of different ways. And like I mentioned, testing specificity is really an open-ended process, and that's really nice. So we can rapidly assess generalizability and specificity across existing data sets. And data sharing is really going to help us do this. So here's one data set that we published. This is Phil Craigle up there in the corner, now a couple of years ago. And it has 18 studies. It's got 3 kinds of pain, thermal, visceral, and mechanical, which are all supposed to be quite different in many ways, and they are different in many ways, and 2 studies of each, and the same number of subjects, 15 subjects per study. So we have 3 types of cognitive control tasks: working memory, response selection, response conflict, and we have 3 types of negative emotion tasks that we picked explicitly to vary across sensory modalities. So we have visually induced sort of pictures, social, which is mostly pictures, and then auditory, negative emotion. And we're calling this data set the Craigle 270 data set. There's 270 subjects, and you can download it, share it, reuse it. It's on NeuroVault, which is a terrific tool, and downloadable directly through the links on our Hanlab that I showed you. And this is what happens when we apply the N to these data. And it gives us, a quite nice and largely specific response to pain, although you can see that there's variability across the sounds, and there are some sound conditions that activate it here to some degree, right? And it gives us a threshold. So this idea, I show this here because this idea of having a set test data set to start evaluating the sensitivity and specificity of models, I think is really helpful. And if you wanted to do this in our tools, then you can use these 2 lines of code, and it will pull the data from the web if you don't have them on your computer already. Okay. So across these studies, we've been able to develop and test what the NPS is really measuring in a way that we could never, we had to do all of these studies. We're not going to do studies on visceral pain in my lab anytime soon, or breathlessness, or all the other possible conditions and cohorts. So to accelerate the pace of science and really put this to the test, it's been a remarkably fun and awesome collaborative process. And this is what we call the psychological tuning curve for this. And this is just simply the set of things that it's sensitive to and specific against, if you will. So it's activated by or sensitive to many types of things that are immediate somatic pain, and possibly some other things as well, like not being able to breathe, this is across studies is another problem that we could discuss. And we're struggling with that and working on it. And it's not activated by a number of other affective conditions that are potentially confusable for pain. And we've started to find some other conditions that do, some somatomotor conditions that actually do activate it to some degree. And that gives us a way of continuing to develop and refine models, right? So if we can falsify the model in some way, then we can keep developing it. But so far, it looks overall quite specific for somatic evoked pain. And if we have a model then that we trust, at least provisionally in some way, then I can start to use that model to make inferences. And I'll just show you one example. In my lab, we've really been interested in using this model and newer models that we've developed to try to make inferences about which kinds of self-regulation strategies work. What about mindfulness and meditation, and what about attentional distraction or social support and having somebody hold your hand? So these are all things that we can sort of compare on the same footing in terms of how they affect these brain measures. And this is just one example from very recent collaboration with Eugene Duff and Rebecca Slater in Oxford. And what they found was that they were able to replicate that the NPS responds to a mechanical painful stimulus in adults, and then in 2 samples of infants where you can't ask them how they're feeling, there is a dose-response relationship, a positive response and a dose-response relationship in those infants. And this is, small detail here, but we divided up, based on work in previous studies, the NPS into sort of 2 parts, the pro-pain, pro-nociceptive parts that you see here in red, and then these areas that are really multimodal, multisensory areas or context-related areas that don't respond to increasing the heat. And these don't match in the infants, but the sort of nociceptive pain regions are activated. So that's just one example. And that can help us to develop an understanding of what the model is measuring and interpret the model. And so I'll just give you a little drop on this. And by the way, I don't know how I'm doing on time. I'm assuming that I have a couple minutes left, so jump in if I don't. I think interpretability is a really important thing to know when a model is going to fail and why it's making the predictions that it's making. And this has been kind of a debate, but I think if these things are going to be useful, then we need to understand what they're actually doing. So this is from Wani's lab as well in a recent paper. It's really a pretty comprehensive take on at least some of the things that might be useful for helping to interpret patterns, both the simple linear patterns that I've just described to you, and also deep learning patterns. They could be other kinds of models as well. And there's code that's now both in MATLAB and Python, which is new to me. To me, Python's still a snake, but I'm going to learn. So then there's feature level assessments of which voxels are important or salient. There's a model level assessment, which includes some of the things that I talked about, its sensitivity and specificity, but also things like which kinds of confounds might it respond to. If you try to use some transformations of head movement parameters or medication use parameters and other things, can you reproduce the model output? And is that a plausible mediator of the relationship between the stimulus that you're trying to study and the model output? Where are its weak points? How generalizable is it? And then a biology level assessment, which has to do with whether you can take those model features and you can relate those to other literature and sort of validate them externally that way. And preferentially, we'd like to validate them using invasive studies where we can manipulate those mechanisms or those brain features and then understand, what are the effects on the phenomenon. So there's different ways of doing that. Some use things like NeuroSynth or now NeuroQuery, which is a recent, sort of wonderful tool that's up there to develop association maps with what other people have studied and the terms they've used. I think we can take those with a grain of salt, but they can be really useful. And also connections with more invasive studies. And this is just an example of a kind of a workflow. And there's a lot here, but you'll be able to see it in the paper and on YouTube. So there's, I don't know if this is a 12-step program. I think so. Step 1, modeling the data. There's model development and assessment of performance. Like, does it actually perform sensitively and specifically? Is it confound free, or can we rule out some potential confounds? And if so, can we start interpreting the features? Do they make sense? Then, is the model generalizable? Is it biologically plausible? And at each point, a model can fail, and you can sort of go back to step one, or even build in more information, about where it's weak into the training process and develop a better model. So then we can start to develop these things and understand how the model relates to other literature, to other brain patterns, to brain networks, and so forth. And the idea of this increasing wedge here is that, the farther we go along this, it's really quite a lot of work. And if we're going to test a model's generalizability, for example, on a broad population, maybe multinational, multi-ethnic, multiracial, then, we're going to it's going to end up costing, hundreds of millions of dollars at the end of the day. And that's what drug companies do every year. And so the idea is we should scale up models in proportion to their utility. A couple other quick ideas about interpreting models. We've had a lot of mileage out of taking these canonical resting state networks, which are vast simplifications of the brain, but then asking, okay, take a model, like a model that's predictive of emotion, and how does it break down in terms of relationships with these networks? And you can see one profile. Well, what about pain, or NPS? That shows a different profile of loading. And what about another model like this vicarious pain model? Again, there's a different profile across networks. So that can give us some insight into how similar these models are, and which kinds of other associations there are. So again, we'd like to encapsulate these common functions in very simple code so that we can have simple workflows to do these things. Here's another one that I rather like, but it's more complicated plot. And the idea of this is to ask the question, well, how much of the brain do I need to make this prediction? And down here, this is pain in one data set with 30 people from our lab. And down here, you see the best searchlight model. This is the sphere at the bottom. And you see that it doesn't predict very well at all. This is the sort of best across the brain in a cross-validated sense. And then if you start using individual networks, the more voxels you include from those networks, the better the prediction gets. And it kind of levels off at different places depending on the network. The dorsal attention network is quite a good one, actually, here. And then you can say, well, what happens if you use the whole brain? How much better can we do? So by starting to make those model comparisons, we can start to make inferences about which bits of the brain are necessary and sufficient to predict a particular outcome. And so I think that's also quite a valuable kinds of process to engage in. And so we've tried to include that in more and more of our papers. And this is the last bit, just to show you that if you take this sort of very complicated multivariate predictive model now from a new study, you can actually look at local regions like the thalamus. And what surprised me in some of this recent work that we've been doing is that the weights, the model weights, i.e. what predicts pain, really varies across the thalamus, even in very systematic ways. So there's a lot of information about structure, structure there. I've just put stars here where the where there are known pain-related processes, and we see the strongest relationships with the model in those regions. Okay. And again, simple code. So finally, my brief coda about model sharing, which is that we have a proliferation of models. There have been several reviews about this. Kyle just did a really nice one, as well as Vince Calhoun's group. And you can see they span the spectrum of disorders, and they're really growing across time. But they have what my colleague Walter Michel used to call the toothbrush problem. Nobody wants to use somebody else's. So we have 600 maps, we have 600 different models. And I just want to make a plug for what we need is sharing and testing of models across labs. And so as you can see here, there's many sort of candidate models, but there's only a few that we found, at least a couple of years ago, that were really tested on independent samples from different laboratories. And some of these things you can see here, like the SPARE AD for Alzheimer's, this is Parkinson's related, and there's this as well. So we really need to have multiple groups, test the same model, validate it, challenge it, and so forth. So I like this idea that's really proliferated of a model zoo in AI. You can take these pre-trained models and do all kinds of things with them. So we started our own little model zoo, and it's got multivariate predictive models from all the things that we've developed over the last years. It's got meta-analysis maps from all the meta-analysis we've done. It's got brain parcellations from our group and other groups as well, and we're happy to include other people's models because a vision for the future really is that we would have this ability to take a data set and apply some sort of robustly validated and trained models. And we can learn something about the models and about our data as well. And if you if you extrapolate this to the clinical setting, ultimately we love to have this case where you do the kinds of brain imaging that we're doing routinely on people and paying them to do. But then this can be interpreted in terms of your risks for health. What's your risk for Alzheimer's? What's your risk for Parkinson's disease? What's your risk for other vascular events and so forth and so on? Are you predisposed to chronic pain? So that if you were to get surgery, would you then be at risk based on your phenotype? And all of those things are, I think, within reach with the direction that we've been heading. And I'd love to see that happen more. Okay, I am done. I talked about these 4 things. And final thoughts for what we need more of, flexible pipelines and tools for interactive analysis, handling between designs, model interpretation and explanation, and model sharing and reuse integrated into software. So let me just thank my lab and collaborators, and thank you very much for your attention. And I'm sorry that I'm over time, but thanks.